In this video, we'll explore the different models of DNA replication. First, we'll cover the role of DNA and discuss the key features of DNA structure and the replication process that informed the proposed models of DNA replication. Then we will delve into the three main models proposed for DNA replication: the conservative model, the dispersive model, and the semiconservative model. So, what is the role of DNA and why does its replication matter? DNA stores the information needed for living cells to function properly and for building new organisms that maintain the characteristics found in their parents. DNA molecules are responsible for transmitting hereditary information across generations and cells. This implies that faithful replication must occur before each cell division. However, faithful replication does not preclude genome rearrangements, which take place during meiosis and lead to the formation of gametes with new combinations of alleles. These rearrangements play a crucial role in the evolution of species and the creation of molecular diversity. Successful rearrangements do not eliminate critical information necessary for the development of an organism. To summarize, then, a successful model of DNA replication should allow for both faithful replication and the rearrangement of genomic DNA. As we explore the features of DNA replication, it is essential to recognize its role in maintaining genetic fidelity while enabling variability. DNA carries genetic information from one cell to the next, so the DNA molecules within a cell must serve as templates for replication. This process must be tightly regulated and occur before cell division. Like any biosynthetic process, DNA replication is not foolproof. Errors can occur during the copying of information. Accumulating errors across many replication rounds could lead to a significant loss of genetic information. Therefore, proofreading mechanisms are essential to maintain the accuracy of DNA replication. Additionally, DNA, like any biomolecule, can be damaged. Such damage can result in the loss of genetic information. To mitigate this, DNA repair mechanisms are in place to ensure the integrity of the DNA and to coordinate this control with cell division. The secondary structure of DNA molecules suggests that each DNA strand can be used as a template during replication. This requires a set of enzymes for strand separation and managing changes in DNA topology. These enzymes also catalyze DNA synthesis, coordinate DNA replication with cell division, and maintain DNA integrity. When Watson and Crick proposed the double helix structure of DNA, the molecular mechanisms supporting DNA replication were not yet understood. The interlocking of the two DNA strands around a common axis presented a significant challenge. It was initially thought that replication would require the complete uncoiling of the double helix, which seemed unlikely given the size of a complete genome. For instance, a small DNA molecule with a molecular weight of 3 million would contain approximately 5,000 base pairs, meaning the DNA strands would need to turn around each other about 500 times to separate. These concerns about the feasibility of uncoiling the double helix led to the proposal of three models of DNA replication: the conservative model, the dispersive model, and the semiconservative model. Each of these models offers a different explanation for how the double-stranded DNA could replicate without necessitating complete strand separation at once. Let's take a closer look at the conservative model of DNA replication. In this model, both strands of the original DNA molecule direct the synthesis of a new double-stranded molecule. The cell produced by the first division contains the parental double-stranded DNA, shown in green. It contributes two cells, one with parental double-stranded DNA, shown in green, and one with the newly synthesized double-stranded DNA, shown in purple. The cells produced by the first division contain the entire parental DNA, which is completely inherited by one of the resulting cells. The newly synthesized DNA molecule, in purple, divides into two cells that both contain this double-stranded DNA. In the dispersive model of DNA replication, the two new DNA molecules are patchworks of parental DNA and newly synthesized DNA. This model does not require the complete uncoiling of the double helix. During the first round of DNA replication, both strands are replicated at the same time, Each original strand, shown in green, is broken at each half turn, and each fragment is covalently linked to a newly synthesized fragment, shown in purple. The linkage occurs between old and new fragments of the same 5' to 3' polarity, ensuring that the information content of the two new DNA molecules remains consistent with the parental DNA. Consequently, with each round of replication, the original DNA content becomes progressively diluted among the newly synthesized DNA. This results in the original DNA being gradually spread out and mixed with the new DNA over successive generations. Now let's explore the semiconservative model of DNA replication. In this model, each strand from a given DNA double helix serves as a template for the synthesis of a new strand. After the first round of replication, the resulting cells inherit hybrid DNA double helices. Each one consists of one parental strand, shown in green, and one newly synthesized strand, shown in purple. Following 2 rounds of cell division, 2 of the resulting cells will each inherit one parental strand, while the other 2 cells will each contain one strand that was synthesized during the first round of replication and one strand that was synthesized during the second round. Consequently, in a population of cells, there are always 2 cells containing one strand from the original cells. This ensures that genetic information is preserved and passed down through successive generations with high fidelity. The 3 possible DNA replication models, conservative, dispersive, and semiconservative, can be tested by tracking the fate of DNA strands through multiple rounds of replication using isotopic or chemical labeling techniques. Meselson and Stahl utilized isotopes of nitrogen, N15 and N14, to demonstrate semiconservative replication. Bacteria were initially grown in a medium containing the heavy isotope N15, which was incorporated into their DNA. Subsequently, the cells were transferred to a medium containing the lighter isotope N14. DNA samples taken after each replication cycle were analyzed via cesium chloride density gradient centrifugation. This approach allowed Meselson and Stahl to distinguish between newly synthesized DNA and parental DNA, providing strong evidence for the semiconservative model of DNA replication. Other methods to trace DNA replication include using nucleotide analogs such as 5-bromouracil or radioisotopes like carbon-14 and tritium. These methods involve labeling newly synthesized DNA during a single replication round, followed by a switch to non-labeled media. In the conservative model, the first generation following the medium switch should contain 50% unlabeled DNA. The percentage of labeled DNA should decrease by 50% with each subsequent round of replication. In the dispersive model, every DNA molecule produced after the medium switch is labeled, but there is a steady decrease in the proportion of labeled DNA with each round of replication. This method allows scientists to distinguish between the models by observing how the labeled DNA is distributed in successive generations. In the semiconservative model, every DNA molecule produced in the first round of replication after the medium switch is labeled. In the second round after the switch, 2 out of 4 DNA molecules are labeled and 2 out of 4 are unlabeled. Because the replication experiments involve cell division, let's pause and refresh our understanding of this process. During S phase of the cell cycle, DNA is replicated, and the amount of genomic DNA doubles. This is followed by entry into mitosis, which marks the beginning of cell division. At the end of mitosis, the 2 resulting cells separate from each other during cytokinesis, and each resulting cell inherits half of the replicated genomic DNA. In diploid eukaryotes, genomic DNA is found in pairs of homologous chromosomes. After replication, each chromosome consists of 2 chromatids called sister chromatids, which are exact copies of each other. During mitosis, specifically in anaphase, the sister chromatids separate from each other. After cytokinesis, each resulting cell receives 1 copy of each chromatid, which ensures that the genetic information is accurately distributed between the 2 new cells. With this information in mind, let's get back to the DNA replication models. In 1956, Taylor, Woods, and Hughes published an analysis of chromosome duplication in fava bean roots that provided evidence that supported one of the 3 replication models. Before discussing their experiment, it's important to understand a few facts about cell division in growing roots. Cell division can be easily studied by incubating roots in a solution that contains mineral nutrients. The cell cycle in fava bean roots is about 24 hours, with approximately 8 hours between the start of S phase and anaphase. The fava beans used in this study had 12 chromosomes. In their experiment, the roots were incubated in a solution containing radioactive thymine for 8 hours, then switched to a solution with unlabeled thymine. This step aimed to label the DNA of the few cells in the root that had undergone one round of DNA replication during the labeling period. Next, the roots were treated with colchicine. This drug blocks the polymerization of microtubules and blocks cell division in metaphase, where mitotic chromosomes are easily visible. In colchicine-treated cells, mitosis cannot proceed, but DNA replication is not stopped. This step aims to make the chromosomes easily visible under light microscopy, allowing scientists to distinguish the sister chromatids. The number of chromosomes in a cell after colchicine treatment indicates the number of replication rounds. 12 chromosomes indicate 1 round, 24 chromosomes indicate 2 rounds, and 48 chromosomes indicate 3 rounds of replication. In the final step, the roots were squashed onto microscope slides, and a photographic emulsion was applied. The slides were incubated in the dark for a period and then developed like a photograph. This process, called autoradiography, visualizes regions of the chromosomes that are radioactive because the radioactivity exposes the photographic emulsion, which makes these regions visible after development. In this experiment, chromosome labeling with radioactive thymidine was carried out for 8 hours. Following the switch to a medium without radioactive thymidine, colchicine treatment was applied for either 10 or 34 hours. The goal was to observe how the number of labeled chromosomes or chromatids changed with varying lengths of colchicine treatment to provide insight into the rounds of post-labeling replication. The left page here shows a diagram of the experiment showing the expected outcomes of radiolabeling chromosomes during replication. The results from Taylor, Wood, and Hughes's experiment showed that the number of labeled chromosomes or chromatids increased as the length of colchicine treatment increased, indicating that the number of post-labeling replication rounds also increased. This confirmed that colchicine does not block DNA replication. For a 10-hour colchicine treatment, most cells in metaphase contained 12 chromosomes, all of which were labeled on both chromatids. This indicated that cells did not pass through more than one S phase during the 18 hours between the start of labeling and the end of colchicine treatment, as there were no unlabeled chromosomes or chromatids. When colchicine treatment was extended to 34 hours, cells in metaphase exhibited several patterns. In the first pattern, there were either cells with 12 unlabeled chromosomes, which indicated that these cells went through S phase after the labeling period and during the colchicine treatment, or cells with 12 labeled chromosomes on both chromatids, which suggests that they passed through one S phase during colchicine treatment. In the second pattern, there were cells with 24 chromosomes, where only one chromatid was labeled on each chromosome. This suggested that there was one round of S phase during the labeling period and another round during colchicine treatment. During the first S phase, one chromatid was labeled, and in the second S phase, the newly synthesized chromatid was not labeled. Rarely, a third pattern was observed of cells with 48 chromosomes. Half of the chromosomes were labeled on one chromatid, and the other half were unlabeled. These cells underwent 3 rounds of S phase, 1 during the labeling step and 2 during colchicine treatment. We can interpret these observations as follows. Cells with 12 unlabeled chromosomes went through S phase after the labeling and during colchicine treatment. Cells with 12 labeled chromosomes on both chromatids went through 1 round of S phase during colchicine treatment. Cells with 24 chromosomes with 1 labeled chromatid went through 1 S phase during labeling and another during colchicine treatment. Cells with 48 chromosomes underwent 3 rounds of S phase, including 1 round during the labeling step and 2 rounds during colchicine treatment. The exploration of DNA replication models highlights the complexity of how genetic material is faithfully copied. The Messelson and Stahl experiment that was carried out 1 year later was pivotal in confirming the semiconservative model, further advancing our understanding of this essential process.